name is Sarah Davis, and I'm a student in Dr. Anastasia Karova's lab, where we study and develop therapeutic siRNAs. This poster shows preliminary data from our lab demonstrating the potential utility of this technology for the treatment of Alzheimer's disease. Alzheimer's disease, or AD for short, is a neurodegenerative disease characterized by memory loss, dementia, and depression. Late onset AD represents the bulk of patient cases, and beta amyloid plaques and tau protein tangles are pathophysiological hallmarks of this disease. This project focuses on inhibiting the expression of tau. To do this, we used a divalent siRNA, which means each siRNA was connected to another by a linker through the 3' ends of their sense strands. The di-siRNA was fully chemically stabilized with fluoro or O-methyl at each 2' ribose position, vinyl phosphonate on the 5' end of the guide strands, and phosphorothioate linkages at all of its termini. The siRNA sequence was selected after extensive screening in Schussy cells. We performed this study using the PS19 mouse model. These transgenic mice express the P301S mutant form of human tau at 5-fold higher levels than the endogenous mouse protein. In these mice, the human tau protein accumulates in the brain with age, resulting in significant neuron degeneration in the hippocampus by 9 months. In fact, the median lifespan of these mice is 9 months. So when we injected them at 8.25 months, they were already very sick. We did a single ICV injection. 5 weeks later, we measured their body weight. Compared to their weights right before injection, the treated mice lost less weight than the mice injected with a non-targeting control di-siRNA. At this time, we also assessed their nesting behavior, which is a natural behavior that declines with age in this mouse model due to cognitive decline. To do this, we separated the mice into individual cages and gave them one pre-weighed nestlet for 24 hours. Then we removed the portion of the nestlet that remained untouched after this time, dried it out overnight, and weighed it. We were encouraged to see that nearly all the treated mice tore up most of their nestlets, which is a stark difference from the NTC group. 6 weeks post-injection, we used a 7 Tesla MRI machine to take live images of the mouse brains. We were very excited to take advantage of this amazing technology for the first time, and we are still analyzing these images. The first thing we did was blindly hand trace the hippocampi. We did this across different sections. Shown here is the data for one of them, where we felt confident in our measurements due to the large area of the hippocampus, which is outlined in green. We saw that treated mice had significantly larger hippocampi than the NTC mice, suggesting that tau silencing by the di-siRNA helped limit its degeneration. The mice were sacked immediately after the MRIs, and their tissues were processed and evaluated for tau, mRNA, and protein levels, and those results are shown on the poster. You can see relative to NTC, average mRNA expression is equal to 26% in the striatum and about 50% in the hippocampus and entrinal cortex. And the protein data shows total tau and phosphorylated tau silencing in the hippocampus and striatum as well. So, as I said, the study is ongoing, but we think that our results thus far, I've shared with you today, are encouraging and merit further investigation of an siRNA treatment for late onset AD. Thank you for your time. I hope you enjoy this poster. Feel free to take your time looking it over and to email me with any questions. Okay, bye.